Okay, well, thank you, Trevor. As he said, I am Morrell Chhay. I'm with the Center of Excellence on Disparities, and today I will be talking to you about variation of mitochondrial DNA and major histocompatibility complex in the Red Hills Roach populations. And yes, we are dealing with fish, not cockroaches, if anyone, it's a common misunderstanding. So to introduce this topic, we will go into where the Red Hills are. It's approximately about an hour away from Merced. It's designated as an area of critical environmental concern, and that is mainly due to the serpentine soil up in the area and the seasonally isolated pools with the Red Hills Roach in it, which is the focus of our talk today. Zooming in, this is a closer look at the Red Hills map. The gray shade indicates the serpentine soil in the area, and we have our different creeks here: Horton, Amber, Roach, 6-Bit Gulch, and Bickabee. And those were where I got all my samples from for the Red Hills Roach. And as you can see on the map, you see it's a nice flowing creek. They're all connected. But right now, the creeks are more or less non-existent. They're dried out. If there is any kind of flow, it's more or less a trickle rather than a creek itself. So what's left is these small, shallow pools with the roach trapped in it. So what does this all mean? The purpose of our study is to look at the genetic diversity of the Red Hills Roach. Since they're seasonally isolated, we want to see how diverse they are, or lack thereof, and to see how they're able to persist into the future. So in order to take a look at the diversity genetically, we have to know what to amplify from the DNA. So we chose mitochondrial DNA, specifically cytochrome B, which is a protein component of the electron transport chain. And the nice thing about mitochondrial DNA is that it has a steady evolution rate, so we can see if there is any interaction going on between the populations. And for the major histocompatibility complex, we have DAB1, UAA, and OL93. And these are just different genes of the MHC. And what we can look at with MHC is if there's any variability going on, we can see how well they can adapt and how well they will do in the future. Once we extracted our DNA to take a look at, we did that through a Quigen DNeasy package. We amplified it through polymerase chain reactions, PCRs, through the certain primers that we had, cytochrome B and the previously mentioned MHC genes. And once we've amplified our samples, we actually sent it out to UC Berkeley to be sequenced and sent back to us. And what we did, we looked at it on a computer with two programs, Sequencer and Mega5. And Sequencer, what we can see from there is if there's any differences or any variations in the sequences of the genes itself. And once we determined that, we managed to put it into another program called Mega5, which creates a phylogenetic tree for us to take a look at and see where they are and what's going on. And because we have phylogenetic trees, we have to have outgroups. The three outgroups we've used for our trees are the Rio Grande silvery minnow, the pit roach, and the speckled bass. The importance of the outgroup for a phylogenetic tree is to have a reference point to see where our samples fall. It's more or less like a frame to a picture. Once we amplified our DNA, we've had more or less complete success with mitochondrial DNA, the cytochrome B. We've lost one or two along the way. For the MHC primers, however, we weren't so successful. For UAA and OL93, we didn't exactly get to amplify anything out of that. So we've mainly focused on DAB1 where we had some success. So we're left with this. Right here we have our populations, the Horton Creek, Amber Creek, Roach Creek. And in the column next to it is what we've amplified for cytochrome B. So for Horton Creek, we had 23 samples, and we managed to amplify 22. Then with the program Sequencer, we looked for unique sequences. We managed to find 7 unique sequences out of the 22 that were amplified. For DAB1, we have our number attempted. We attempted this sample amount, and we managed to amplify 4, and we found 1 unique sequence. Once we managed to amplify and program everything, we've created a phylogenetic tree, which we have right here. Our outgroups, the speckled bass and the pit roach, are right here, and the Red Hills Roach are all along this clade. What's interesting is you can see that there's interaction going on between each subpopulation. because when we suspect that each population is secluded and isolated from each other, they should be each in their own little clade. But that's not what you see here. You see that there's interaction going on, there's mixing, and even more interesting, Horton Creek and Becca B over here, which is labeled ATT, they're even off in their own little clade, slightly far from, just slightly from the Red Hills Roach clade. So what we can tell from this for sure is that there is gene flow happening. There is interaction happening between subpopulations, and that there is variation that we see for the cyclone B at least. For our DAB tree here, side note, this is not a phylogenetic tree. This tree is mainly to show where our Morrell Chhay Roach samples fell under for the DAB1 primer. A previous study had amplified the Rio Grande Severe minnow, and they managed to sequence it and organize it on a phylogenetic tree, where they found 4 gene copy variations of the DAB and MHC gene. They found DAB1, 2, 3, and 4. So once we've created a tree for that, we put in our samples, and we managed to find that the Red Hills Roach falls under the clade of the DAB1, indicating that what we did amplify was DAB1. Now for the samples that did not amplify with the MHC gene, we can say 2 things. It is possible that the DAB1 primer that we had used that worked for one species did not work for our Red Hills Roach, meaning there was not enough generality within the primer. So the primer was not even able to latch on and amplify the DNA in the first place. And another possibility is through gene copy variation. As you saw, the DAB that I had amplified was just DAB1. It's possible that the other samples that did not amplify were either DAB2, 3, or 4. So what we can conclude for sure is that the Red Hill Roach population, there is gene flow happening, as we saw from Cyclone B, and there is interaction going on between the subpopulations. For the samples of DAB1, we can conclude that it is for sure that what we amplified is a DAB1 gene. And for the samples that did not amplify, we are leaning towards the matter that it's due to high gene copy variation, which leads me to my future work. Throughout the year, I hope to sequence MHC genes with different primers to get a clear picture of the MHC genes, see if there's any gene copy variation, and see how many there are. And from there, we can infer how they can persist into the future, if they can adapt to, say, a new pathogen being introduced into the environment. And from the Cyclone B, we know that there's gene flow happening. So we want to take a closer look and see who is moving about. And through this, we can do that by analysis of a hypervariable repeated sequence, more or less microsatellite genotyping. The nice thing about this is that microsatellite genotyping is highly variable and is special to each sample within the populations. So we can see who is moving about and who is staying and what exactly is going on. Before I move on to questions, I would like to acknowledge the members of the Aguilar lab and for their support, and of course the Center of Excellence on Health Disparities for giving me the opportunity to review research with the Aguilar lab. Are there any questions? Yes?
Morrell, what's the significance of the serpentine soil?
The serpentine soil, what's interesting about it is that it's high in iron, hence why it gives it that the red clay look, and hence the red hills. And more or less it affects the plant life. It gives it a very unique plant life about it.
And also, when the rivers are connected, because the water level's high, do those roaches move around or do they stay in pools?
That's what we're trying to see with the microsatellite genotyping, yeah. We know they interact, but we're not sure how they interact yet. So that's something we hope to find out later on. Yes?
I'm interested in your sampling. Were you able to go there and you sampled yourself and maintain your samples?
No, the samples were provided me from before, but however, I did go up there to take a look, mainly just to get an idea of how shallow those pools are, and it really is a shallow pool. You can step in, it's probably up to your ankles or up to your shins at the very most. Yeah. Yes?
Do you think the implications of this are for sustainability or for health or for both? I'm trying to understand what the implications are for health. I can understand for sustainability.
Well, definitely for sustainability.
Yeah.
Because we have to know, well, what's going on with these populations before,
right.
How long they will exist, or if we plan on doing something, we should know before we even do it.
Mm-hmm. Do you think there's, I mean, what sort
For
of,
sustaining to health, I'd have to say I don't know. I can speculate and come up with something, but that wouldn't do it justice.
are the sequencing processes the same for any type of DNA? I mean, in other words, is it unique? I'm totally ignorant on this. So is it unique to the species, or would you be using the same type of sequencing for other
You mean the
DNA?
process of actually sequencing the
Right, the
parts?
actual process, is that species unique or not?
In the sense of doing the process, you can apply that to other different species.
Uh-huh.
Are you asking if this roach is a unique species?
No,
Oh,
I'm
okay.
asking if the process,
Yeah,
is-
the
the
process you
process
can
is the
apply.
same
Yeah,
regardless of
regardless.
what type of animal you're talking about.
Yeah. Of course, you have to change small things, but generally, yes, it's more or less the same. Yes.
So from a health and nutrition standpoint, do you know what kind of levels of omega-3 fatty acids are present in the roach fish farm?
I do not know that, actually. I haven't met anyone who has eaten the red roach. I don't even know if we can eat them. I don't think you want to. They're very, very small. Maybe as an hors d'oeuvre. Yes.
And you mentioned you're trying to figure out how long they're trying to exist, how long can they exist in the condition they are, and what you can do to make them exist longer. Is this in the genes or in the environment?
It's a play on both. We're definitely focusing on the genes. See, for the MHC, which deals with the immunology and all of that, there's high variability. We know that they can adapt if there's a new pathogen. I'm not saying that there will be a new pathogen introduced, but if there is, then they're more likely to adapt if they have high variability, because they have a lot of things to adapt to.
So you're just observing it. You're not actually doing anything. Or?
Yeah, we're just observing. We're not going to introduce a new pathogen and see what happens.
Yeah, I understand that, but not in that sense. In the sense to the actual, like, to the species you're researching. Like, there's no way, of course, that you can like help out the genes or anything like that, right?
What do you mean by helping out the genes?
I don't know.
Talk to me later. Go see my proposal. Are there any other questions? Yes, Chris?
You said that the pools are really small.
Mhm.
So, like, how many fish would be in each pool? Like, when it goes to be located?
I'm not willing. I don't know. Saying there and counting, there's. What's your definition of a lot? You know?
Because
Yeah.
they're their own population. So I was just wondering what the population.
Yeah, the
size is.
population sizes are relative to what pool they're in.
Mhm.
So I didn't go up there and sit and count each one. So
Yeah.
I can give you an idea. It's small. There's
Okay.
small populations.
Okay.
So it's not. They're not jumping out of the water. They have enough room to swim around and get away from us.
Okay.
Questions?